lessons on the basics of their disciplinary perspectives and approaches, just to help us see the world through their lens within the context of coastal issues, so that we can all build a shared set of lenses that we can use to hopefully bring different and new foci into our research into the future. We have two 20-minute lessons with a good amount of time for discussion in between. Today, Dana Hunt and Angela Harris will be our instructors, and thank you very much, Dana and Angela, for your service on this account. And so at the end of the first 20 minutes, we'll have 10 or 15 minutes for discussion. Catherine and Arde will do the moderating of the question and answer. So she'll let you know over the chat, give you a reminder to start maybe typing in questions. You can either have Catherine read them out in the end, or you can volunteer to ask them yourself. And any other business I should cover, Catherine?
That's good.
All right, then first I get to introduce Dana Hunt, who's going to kick off here, and you can go ahead and share the screen if you'd like, Dana. I don't think that I've got your proper title here, but Dana got a pair of bachelor's degrees from Rice in biochemistry and environmental engineering, and a PhD from MIT in engineering and civil engineering, I believe. And she studies microbial ecology in estuarine settings. Take it away, Dana. Thank you.
So today we have our microbiome and engineering background, at least double header, although I think I am officially more of an ecologist today. So I'm going to kick us off, and I wanted to take you guys kind of into the microbial realm. And if anyone notices there were some issues with the sound before, let me know, and I'll turn off my video. So if you go out, this is the ocean right behind me here at the Duke Marine Lab. And if you go out there and take a teaspoonful of seawater, you'd find about 1 million cells. And that's pretty much true no matter microbial cells. And that's pretty much true no matter where you go in the coastal ocean. There's a little bit less in the open ocean or deeper out there. But pretty much the vast majority of life on the oceans, and indeed on our planet, is microbial. And it's not just that there's one or two things out there, there's a lot of different things out there. So not just the vast majority of cells, but the vast diversity of life is really microbial. So if you went to school 1 million years ago, like I did, you probably learned the 5 kingdoms of life. So animal, plants, and fungi, and so I've kind of circled those groups. And then there were these other kind of random groups, the Monera and the Protista, that kind of got ignored because there were small things you couldn't really see. But those are actually the vast majority of life. So all these other groups are those tiny things that you can't see readily with the naked eye. And I'm really trying to take this at a basic level. What I'm going to introduce what a phylogenetic tree is. This is the last time you'll see one in my talk, hopefully. But I just wanted to highlight that when you look at this tree of these different groups, the longer the branches, the more distantly related those organisms are. So it's not just that there's a lot of different things that we can't see. They're also incredibly diverse, and they've been deeply branching from one another. So the first microbes evolved on our planet about 3.5 billion years ago. And it's really common to think of them as to continue as a primitive single-celled small thing because we can't see them. But these microbes have been evolving over time, evolving with us, starting to deal with things that we do and degrade our pollutants, adapt to new environments, and changes over this long evolutionary timescale. So later today, Angela's going to tell you about all the nasty things that microbes can do to you. But I'm going to really emphasize that microbes were there first, and they inhabit our oceans and perform essential ecosystem services that we often kind of ignore. So microbes really drive marine food webs. So they, again, are microbes, small things we can't really see. We can't tell who they are just by looking at them. And we use a range of molecular methods or sequence-based methods to tell what they are. I'm not going to go into detail today about those. I'm happy to nerd out later if anyone wants to talk about those with me and how we determine. So I'll try to refer to them as taxa or types, but you can think of those as equivalent to maybe species in groups you're used to thinking about. So what do these microbes actually do in marine food webs? So marine microbes are the base of marine food webs. So I've circled here the large and small phytoplankton. So we can think about those as the single-celled plants of the oceans. So they're taking up carbon dioxide from the atmosphere and harnessing energy from the sun and generating new biomass. So that goes to support higher trophic levels. So you got into the open ocean, there aren't any plants. There's just these single-celled microbes. It's more mixed in coastal systems. And then I also wanted to highlight these uncharismatic black dots, even on this figure, and these are the bacteria. So what do the bacteria do? They actually have diverse functions, but today I'm going to emphasize them as the single-celled heterotrophs or animals of the ocean. So they are going to eat all of the detritus that are produced and waste that are produced by our phytoplankton, as well as our fish and other organisms. They're key to controlling where carbon goes in our ecosystem, whether and nutrients as well, whether it gets recycled in the ocean and made available for use, recycled as carbon dioxide and inorganic or organic nutrients, or whether that carbon gets sequestered into the deep sea. So they're really kind of the gatekeepers of what happens to a lot of the carbon in our system. So humans have the wonderful potential to upset a lot of these ecological balances. So one of the common things that we think about when we think of how we upset microbes is through eutrophication. So as human activities add nutrients such as nitrogen, phosphorus, and silicate, that's really taken up by our phytoplankton, our single-celled plants. They use that to generate biomass, and that can lead to problems. So one aspect of that are red tides, seen on this image over here, or harmful algal blooms, which can impact both human and environmental health. But as those cells sink, or as they're consumed by organisms and they sink to the bottom, our bacteria chew them up, they use up all the oxygen, and that can lead to low oxygen zones and things like fish kills. And so humans have the potential to kind of upset these environmental systems. So when I was thinking about topics, obviously the topic of microbial diversity is a huge one, but I really wanted to focus on concepts of disturbance, especially as related to seacoast. So in microbial ecology, we think about two different aspects of disturbance. So one is what I'll call a pulse disturbance, and that's something that happens over a relatively discrete period of time. It alters the environment. We can then assume our microbes change somehow. Over time, they may go back to where they were. I'm being very vague about what these terms are. I mean, you can think about them going back to what they were before, or they may enter some sort of alternative stable state. So if you think about ecology, an alternative stable state on coral reefs may be between coral reefs and fleshy algae, right? That once it's entered this state, it's really hard for it to go back. So an example of we might think of is a pulse disturbance is something like a hurricane. And so that changes a huge number of environmental variables at one time, alters the environment, and at some point we think that the microbial community would probably go back to normal. But humans are also really great at introducing what's we call a press disturbance. So that's a disturbance that happens over a long period of time. And then does the microbial community somehow adapt or evolve? And I'm using the term microbes here, but these concepts are common in ecology to lots of different types of organisms. So again, we have our alternatives of evolving and maybe going back to the way it was before, or again, an alternative stable state. And so a great example of a press disturbance is I'm showing the ocean acidification, or the pH at Station Aloha in the middle of the North Pacific subtropical gyre. You can see there's an annual trend in pH of the system, but then kind of a gradual decline over time. And so how do organisms respond not to something that goes away, but something that persists and continues as kind of this press disturbance? So today I'm really going to focus on the pulse disturbance of a hurricane. So I think it's relevant to a lot of the researchers here and how a lot of us think about microbial systems and our systems in general in Eastern North Carolina. So I wanted to broaden what I'm talking about beyond my own research, but also kind of tie it to things I know. And so I really wanted to focus on hurricanes in the Albemarle-Pamlico Sound system. For those of you not from the area, the Albemarle-Pamlico Sound system is the second largest estuary on the US East Coast. It's a lagoonal estuary. So because it's bounded by these outer banks here, that means there's a long residence time. The water sticks around for a long time in that system, often referred to as a bathtub. It sloshes back and forth, and it only gradually kind of leaks or exchanges through a limited number of inlets. So what that means is that anything that happens in the watershed, the entire watershed, there's 6 river basins that feed into the system, affects the downstream sound system. And as we will learn from kind of foundational work by Hans Perl and colleagues through the Modmon and Pamlico sound system monitoring, is that really the effects of hurricanes can last for a year or longer potentially in these systems. So I really did want to acknowledge the really long-term, multi-decadal time series run by Hans Perl and colleagues at IMS down the road from me in the Neuse River estuary. So the Neuse River estuary is strongly impacted, as I said, by things that go on upstream, but it's also exchanging with this Pamlico sound system. And so this has really been one of the systems in which we've learned a lot about how hurricanes impact microbes as well as environmental variables in coastal systems. So in contrast, my research is actually down here in the mouth of the Newport River estuary based at the Duke Marine Lab. We've been going on for now 11 years of weekly sampling, so not up to Hans's record, but it serves as a really nice contrast. So our estuary is tiny. It doesn't have a huge watershed, but interestingly, it's connected to the Neuse River by a series of canals, human dug canals that served as a navigational aid. So we're actually getting some of the water from the Neuse River estuary. I think it also serves as a nice contrast of what the Pamlico Sound might become as the Outer Banks, additional inlets start to open in the Outer Banks, or the islands start to move and degrade and there's more water exchange. So, so I'm going to try to highlight my work with the PICO logo and Hans's work with the Modmon logo, but I'll try to clarify, but please let me know if there's any questions with that. Okay, and so I think another thing that those of us who live in the coastal zone, and I didn't really appreciate it until living here, is that hurricanes are not all the same. So North Carolina is crisscrossed by a huge number of hurricanes, but as anyone who's lived through one has known, they have different flooding potential, different rainfall, the wind from different directions that causes water to move inshore or move up. And so each of them, they're not purely replicates as we might want an ecology of one another. And so I think the power of these long-term time series is really to provide context for understanding disturbance-related changes or these hurricane-related changes in the context of annual cycles and in the rare non-hurricane impacted year. And so it really allows us to establish baselines. So what do we see with hurricanes? So again, at our PICO site, we see some of the most extreme values we ever see in our time series with these hurricanes. Despite the fact we're right at the Beaufort Inlet, we have very salty water. It's usually 33 ppt or higher, so coastal seawater strength. When we have a hurricane come through, this is Hurricane Irene in 2011, we see a drop in pH, a drop in dissolved inorganic carbon or dissolved carbon dioxide, and we see increases following it in our phytoplankton. So those single cell plants are actually taking up nutrients that get washed down in the estuary, and they tend to bloom following the hurricane. And similar things have been observed in the Maudemont time series, but there's also some unexpected things that I discovered when I was doing additional reading for this presentation. And one of which was that hurricanes can actually have positive impacts on water quality. In this case, the Neuse River estuary frequently has hypoxic events in the summer, but hurricanes in this case seem to act as mixing events that actually increase the bottom water oxygen, making it a more healthy environment, say for fish. And so we can see these shifts in environmental parameters that can persist in some case for months to almost a year, especially in that lagoonal estuary system. So how long do these persist? So we used similar analyses that Hans did to actually look at when environmental parameters kind of go back to normal, and that's indicated by these black lines here. And so Hans's research looked specifically at organic carbon, and they found at our downstream stations, they saw deviation from normal of up to 200 days following a hurricane, so increases in organic carbon. In contrast, we found shorter time intervals, more on the 40-ish days following a storm. This is measuring suspended or dissolved carbon dioxide. When we started to look at biological variables, so again, I said that chlorophyll or phytoplankton tend to bloom following storms. We actually didn't see, it wasn't statistically significant. So even though we see these blooms happening, there's so much biological variability in the system, we don't actually see them as a statistical outlier. So we can't necessarily quantify all the things that are going on. So I found that really, really interesting and starting to do a more statistical analysis. And I think one of the most striking images for me following Hurricane Florence was this dissolved organic matter, the CDOM or colored dissolved organic matter from swamps, blackwater swamps leaking out of our estuaries into the coastal ocean. So how does hurricanes change the biogeochemistry? So again, work in the Neuse River estuary has actually suggested that it can convert this estuary from a net carbon dioxide sink to a carbon dioxide source, meaning that instead of taking up carbon dioxide from the atmosphere, it's actually producing or releasing more back into the atmosphere. And so these disturbance events can have profound changes on the metabolic potential of our microbes. Interesting, so we've done some work, again, as I said, looking at hurricanes. So if we look at environmental variables at our time series, we can see hurricanes are extreme outliers outside of kind of our normal seasonal variability. But when we look at the microbial community, this is following, again, Hurricane Irene. We haven't fully analyzed all the other hurricanes we've had recently. The microbial community is not an outlier. We see outliers are big changes in the microbiome between our weekly samples other times, but not following the hurricane. And I think this is a really great reminder that microbes don't necessarily see the same thing as a disturbance that we do. So I think that's something that most people would categorize as a disturbance. In fact, we were very surprised to see this data. But I think it's a good reminder that environmental context and how what types of environmental variables change actually do matter. So it isn't that nothing changed in our time series. We did see following the hurricane, we saw blooms in some of those single-celled phytoplankton. And when we used a Bayesian modeling approach, and even excluding the year that we had that hurricane, Hurricane Irene, we can see that the abundance of these phytoplankton is linked to things like high nutrients and low salinity. And so it looks like these are kind of organisms that are poised to bloom, and when they're given that extra boost of nutrients caused by or resulting from the hurricane, they do bloom to the highest levels we ever see. But if you look at other years in our time series, they are pretty abundant. They're already there kind of in the fall. So they're kind of as I said, poised to take advantage of these increased resources. So I wanted to start to wrap up. So how can we start to understand all the different things that happen? So I think most of us have thought about hurricanes, or if you think about hurricanes at all, they're what I call multi-factor disturbances. It means not just one thing changes. It's not a temperature anomaly or a nutrient pulse. It's a lot of things change at once. And so can we use manipulative experiments to really understand what the drivers are of hurricane-associated changes? So the nice thing about microbes is that we can take liters of them and put them in a bottle and manipulate them and see what happens. So it can't do that as easily with a lot of other organisms. So we did this previously based on data from our time series. We found that there's, again, these are different colors or different types of microbes, and these are the year-over-year temperature and light patterns. And we saw there was a really strong seasonal pattern in our microbes. But because a lot of things change over a seasonal cycle, we couldn't necessarily assign the source of that change. So we tested temperature because our instinct was that temperature was the strongest driver. So about a 20-degree annual temperature change in our time series. And then we wanted to look at what a 3-degree temperature change would cause. So 3 degrees is the projected kind of long-term global surface seawater temperature increase in the next 100 years. And what we found was that we could actually see the microbiome change over just 5 days in the laboratory in response to 3 degrees of warming. And so that tells us that we were probably observing a real thing in the ocean. In contrast, we also tried ocean acidification and didn't see any statistically significant impact of that. So it's possible there is, and we didn't detect it, but I think it's a technique that we can start to use more of to kind of iterate between environmental observations and experimental testing to help answer questions about future conditions in the oceans. And I wanted to end with just thinking about some questions that we might want to discuss or think about how ecosystem changes, how those might move on to other trophic levels. So again, these are our foundational species in the ecosystem, and how long-term changes may interact with disturbances to kind of act as multi-stressor events to change things. So I am running short on time, but I really focused here on microbes. Sorry, but I did want to highlight or bacteria and phytoplankton, but I did want to highlight that there's a lot of other groups in the ocean. So again, this is our time series, and each of these lines here is just a different organism. And so here's our bacteria and our phytoplankton we started out studying, and they display patterns of almost switching between summer taxa and winter taxa. So then we looked at fungi in the ocean, so our same time series samples, and we saw really different patterns. And those tend to be more episodic, to appear and go away, but they reoccur annually, suggesting this isn't random. And similarly, there's this understudied group of Labyrinthulomycetes, which is another heterotrophic or animal-like organic matter eater, and that displays yet a different pattern. And so there's really a lot going on that we really don't know that much about in the coastal ocean system. And I wanted to thank you guys for your time, acknowledge where I am right now in beautiful Beaufort, North Carolina, and of course, our sampling site right here, as well as a lot of contributors. I think we've had over 60 people contribute to our time series, ranging from undergrads to faculty. Thank you.
Thank you, Dina. While we wait for questions to trickle in, we do have some time for questions. So please put your thinking caps on and put some things in the chat. I have a question for you. Early in your talk, you mentioned the potential for more inlets opening in Pimlico Sound. And as a coastal geomorphologist, my ears perked up. And I'm wondering, do you think the addition of more inlets in Pimlico would influence more the pulse stressors or the press stressors for the lagoon ecology?
Yeah, I hadn't thought about the press stressors, but I've definitely thought about the pulse stressors. So work as by the Hansa sampling team has really found that some variables change from months to a year, right? And so the more exchange you have with the coastal ocean, the more that is going to get diluted out more quickly. So right now, I think a lot of that organic matter and nutrients are being kind of recycled in that really shallow estuary. So organic matter and nutrients come in, it forms biomass, it degrades, it fuels new biomass blooms. And so we're going to kind of retain that. And I really do think that transfers to higher trophic levels. So there's a lot of fish, juvenile fish, that use the area, especially things like menhaden that are filter feeders and actually filter phytoplankton directly as their food source. And so I really think kind of the fisheries of the East Coast really rely on these estuary systems that have kind of reduced, I would say, foraging costs for organisms, because the more easy it is to encounter food sources, the more concentrated it is, the more energy you can devote to growing and the less you have to devote to searching for food. So on the press disturbances, that's an interesting question. I'm not sure I know the answer to that. I think definitely if there's increased kind of freshwater discharge, that'll continue to impact things. Could I call him
That's great. We have a raised hand. Grant, do you want to unmute yourself?
Yeah, I'll even try to turn on my video while I'm doing that. Dana, thanks for the talk. That was great. I was intrigued by one of the comments you made about the positive impacts of hurricanes. You don't never normally think about them that way here on the coast, as you know. And I think you referred to sort of clearing up, as I understood it, hypoxia events in rivers. Can you tell me a little bit more about that? Like, are those hypoxic events that are caused by other anthropogenic activities? So that's one question. The other is, are there other positive biological impacts of hurricanes that you're aware of?
Yeah, thanks for that question. So hypoxia events are fueled by a number of causes. So one is nutrients coming in. So that can actually be worsened by hurricanes. So it's somewhat surprising they actually reduce it, but it's actually the wind-driven mixing and I think the reduced residence time. The water's moving faster through systems. So those kind of hypoxic events are really common in the Neuse River estuary in the fall and the summer and fall. So similar to the Gulf of Mexico dead zone or low oxygen zone, that kind of forms every summer and then degrades over time with wind-driven mixing, actually mixes oxic surface waters with those deep oxygen depleted waters. So what are some, is that enough on that or other thoughts? Sort of
yeah, that's helpful. I mean, I was kind of curious if they've always been there, the hypoxia events in the Neuse, or is that like farming and agricultural activities, much like it is on the Mississippi? I know it's a little bit outside of what you're studying, but.
Yeah, so my intuition is that there's always been lower oxygen levels in bottom waters. I think that's true. There's completely natural low oxygen areas. I think that it's exacerbated and increased the impact because of anthropogenic non-point source pollution through use such as fertilizers and human and agricultural animal waste that washes into the estuary. So some other, I think you also asked about some positive impacts of hurricanes. So actually those of you in the triangle rely on surface water. And I think there's a study by some of our colleagues, I think at UNC and Duke, that actually found that the surface water reservoir system actually requires storms or tropical cyclones to actually supplement the surface water, drinking water. My personal view, and I don't have great data to back up this, is that some of the productivity of these estuaries is caused by these kind of pulse events that wash down a lot of nitrogen, and then it's recycled in the system. And so even though it may cause maybe negative short-term changes, it's sort of built into the system that you have this increased deposition at some rate. Does that make sense? That it's normal to have disturbance at some rate. You can think about like fire adapted ecosystems. They can be catastrophic, but they're also part of the reproductive cycle of certain organisms.
That's great. Thanks.
Okay, I think we have time for one more question. Laura, do you want to ask one of your questions? Wait, there we go.
I can do that. I guess I had just following on what Grant inquired about. I mean, if hurricanes become more severe or more frequent going into the future, would you expect the benefit to continue to be positive, or do you think there may be a threshold for positive impacts? And I guess just also related to that is going forward in time, given what's expected to occur in terms of changes to press and pulse drivers, do you have a sense of what the greatest impacts to the microbial ecosystem in the coastal ocean might be, given what you know now?
Okay, that's a lot of questions. So I think in all cases in hurricanes, it's a change to the system. And in some cases, I think at least the microbial residents don't care that much, but other organisms might. For example, in Hurricane Florence, huge swaths of oysters were lost because the salinity changed. So certain organisms may have tipping points beyond which they can't survive. And so I'm not sure we have a great benchmark of where all those tipping points are for all the different organisms and ecosystems. But I think they probably vary, and it may be, as I said, our hurricanes are multi-stressor things. So maybe for oysters, it's salinity. For oyster spot, maybe it's actually pH. For microbes, it might be outcome competition because of nutrients. And so I think there may be different organisms that respond to different aspects of hurricanes. So I think every hurricane has, for different organisms, positives and negatives. And it's really understanding the balance and interplay of those. I think a lot of marine organisms, the stress point will start to be salinity for a lot of things, because most of these organisms have obligate requirements for salt and can't survive even kind of brief forays into very low salt. I didn't mention it, but we've been looking at those microbiome disturbances in our time series, events that changed the microbiome. As I said, the hurricane was not one, but those were temperature extremes. The ones we can identify were temperature extremes or predators that seem to appear. And so there are different kinds of, if I'm thinking about kind of press disturbances overlying, or pulse disturbances overlying these press disturbances of climate change, I think temperature anomalies will be a big one into the future, for at least for microbes. So I think there's still a lot of questions. In spite of all the work we've done to study them, there's always a lot of questions to answer. Thank you.
All right. We have to move on to Angela. Thank you so much, Dana. And Brad, do you want to, oh, you are muted.
I have the power too, apparently. Yes, thank you, Dana. And next in the tag team, Angela Harris, who got bachelor's degree in chemical and biomolecular engineering from Georgia Institute of Technology, and both master's and PhD in civil engineering from Stanford, and who's now at NC State. There you are, Angela. Thank you.
Great. Are you seeing it in presentation mode, not the, okay, great.
Yes.
Thanks everyone. Thanks for this opportunity to chat and to share about some of my work. And, I think it was a really nice coupling to go with Dana to kind of think about microbes in many different ways. I always make sure, so in my work, I really focus on the microorganisms that are harmful to human health. But I always make a point, especially with my 5-year-old, to make sure she's aware that not all microbes are bad. And so it is, I do appreciate this kind of pairing of talks. But I am going to be discussing my microbial water quality and particularly thinking about microbes that can cause infectious disease in humans. And I am focusing on the impacts of extreme flooding events on surface water quality and the microorganisms that we're finding in surface water. And I'm going to be presenting some results from some work that I did in North Carolina as part of a larger team after Hurricane Florence. And so just, this is an image of Hurricane Florence making landfall. It made landfall on September 14th, and it was a Category 1 storm. And when it made landfall, it continued to weaken, and so its forward motion started to decrease, and so that allowed it to drop, much larger amounts of rainfall. And this is, this phenomena of, large amounts of rainfall, slower moving storms is, something that we're seeing much more of and it's anticipated to increase due to climate change. The recent events that have been hitting Louisiana and Texas are kind of testament to this. And when we after Hurricane Florence, there was a lot in the news about, what is the environmental impact going to be of the storm, and really a lot of concern about potential sources of contamination to surface waters from, our industrial agriculture operations, so CAFOs and the hog lagoons becoming inundated, and also municipal waste, so how human waste is being managed and how that might lead to untreated sewage entering our environment. And so there were a lot of these concerns, and they were making headlines, and really trying to understand the extent to which this was an issue. How did it spread? How did the rainfall patterns maybe contribute to the spread of contaminants? I do want to recognize, and I think this was touched on earlier, right, that these are very complex systems, and the impacts of an extreme rainfall event can persist to up to a year, and how your system is going to respond to an extreme rainfall event is impacted by its prior exposure to rainfall as well. There are also many different sources of contamination. When I'm thinking of microbial contaminants that can be causing inputs of pathogens to the environment, we have agricultural and livestock runoff, urban runoff, septic systems, and these can lead to, different exposure pathways after an extreme flooding event as well. And the work that I did, some of the work that I've been involved with does involve understanding human behavior after these extreme flooding events, their perceptions of risks and what their concerns are, so that we can better make sure that we are providing data to people that are in these stressful situations after an extreme flooding event. So as I mentioned, there can be many different types of sources of contamination to environmental systems after an extreme rainfall event. And when I think about these sources and the types of microbial targets that I'm most concerned about, I really think about fecal contamination and sources of feces into environmental waters. And why I'm so concerned about feces is because there are many different types of feces, or many different types of pathogens that can be found in feces. And Dana, talked through the tree of life and the diversity of microorganisms. And there are a range of pathogens from prokaryotic organisms, to viral, to eukaryotic organisms that all can go on to cause human disease and infection. And so there are an array of pathogens that we have to worry about, and their behavior in the environment does vary in terms of their persistent, how they adhere to different particles, how they behave when they are in water versus soil or sediment. And so the diversity of these targets does make it, it can present a challenge in terms of wanting to characterize the risk in surface water quality after an extreme event. These pathogens and feces are transmitted via this fecal oral transmission pathway. This is, in my area of research, we call this the 5F diagram, and you have fluids, fields, flies, fingers, and food that can help aid in the transmission of these organisms. And so when we think about different ways to intervene, so this is a figure that really looks at different intervention strategies. So a lot of my work is actually done in low and middle income country settings. And so some of these sanitation infrastructure options aren't necessarily ones that are being considered in the US, but you can imagine that there are different strategies of where you would want to intervene to prevent exposure to these pathogens. But the strategy that can be really helpful for preventing contamination is managing the waste in the first place so that it doesn't enter the environment. So you're not as much relying on kind of point of use methods for treatment and intervention. And when we think about pathogens and feces that are infecting humans, yes, human feces is quite risky to humans. They can contain, high concentrations of pathogens. So in sewage, you can find very high concentrations of pathogens that cause infectious disease. But this is a study that actually, it was focused on recreational water quality and looking at the health risk associated with feces from different animal hosts. So, on the x-axis here, you have these different animal hosts with raw sewage and secondary effluent being human sources. And you can see the different bars are different pathogens in particular that are attributing to a probability of illness, which is on the y-axis. And you can see this was a modeling study. So using kind of secondary data to estimate the risk associated with different host species. And you can see that different hosts kind of carry different risk. Their feces carries different risk to humans in terms of the zoonotic pathogens that can be found. And cattle, based on this modeling effort, can have similar levels of risk if you're kind of using a indicator organism as kind of a normalization metric for quantifying the fecal contamination in water, but that there are risks associated with pig and chicken feces and even gull feces as well. And due to this risk that is posed by feces, there is much effort and safe management of feces. But we do see that there are large amounts of feces that are produced from humans, from animals, particularly in industrial agricultural settings. We have strategies that have been used to manage feces, but I would argue that there has been some path dependency in terms of which strategies we are employing. And there has been pushes to innovate in this space. And many people do research and waste management processes, but I do think that there has been a growing recognition given the changing climate, given these changing climate patterns, that we might need to rethink how our infrastructure is designed, and that there is room for innovation in waste management strategies. And since our kind of current infrastructure doesn't seem to be doing the best job in response to some of these changing climates, that we do observe substantial loading of fecal contamination in our environment and in waterways. And this then carries this risk of GI illness. And there have been studies that have demonstrated increase in symptoms of diarrhea after extreme rainfall events attributed to hurricanes. So my work really focuses on water quality, and in order to understand, what's going on after an extreme rainfall event, or how our surface waters respond to different rainfall patterns, we have to measure, do some measurement. And so I want to just review some of our options when it comes to measuring microbial water quality, and to kind of discuss some of the strengths and limitations of these different approaches. So, if we're most concerned about pathogens, that is often what we want to target in our measurement. Is let's actually just go after the pathogens that can cause human illness, to understand how they are being influenced by different climatic variables. But there are many challenges with direct measurement of pathogens. They are typically found in low concentrations in the environment. And so you have to have different techniques to really concentrate your samples. And then because of what's in your sample, that can lead to different inhibitors being concentrated as well. And so that can then impact your analysis methods that you might be using, either culture or molecular methods to measure the pathogens. There are also many pathogens to target, and we're also learning about emerging pathogens of concern, right? And so when you're trying to just directly measure pathogens, you run the risk of missing the full picture of risk. It can also be quite expensive to do direct measurement of pathogens, and so that can be a challenge for some of the for research approaches if it's just not in the budget, and for also monitoring efforts, like routine monitoring efforts, that they are quite expensive, and they can take a lot of time to test. Some of these methods to test directly for salmonella, for instance, it can be a 7-day process before you have confirmation that you had salmonella detected. And this typically involves skilled technicians to test, and advanced laboratory settings that can handle these increased biosafety hazards of especially for culturing the pathogens. However, there have been innovations in direct measurement of pathogens. And so in my lab, we have a piece of machinery that now, using molecular methods, we can test for a range of different pathogens in one go. So like, we process the sample once, and we can have 20 to 30 pathogen targets that are being tested in that single sample run. And the costs have come down from what it would have taken if we had done it many years ago, but it is still not, it is still a relatively expensive, on the order of like $100 to $150 to test for this panel of pathogens. But we have seen an uptick in using this technology to evaluate, the pathogen specific incidents in these environmental samples. Due to these challenges, though, often what you will see in the literature are indicator organisms for indicating an elevated risk of microbial contamination, of pathogen presence. And ideally, an indicator would be low cost, easy to measure, timely, like we'll have a quick response time. It correlates well to what we care about, pathogens, but we're also ultimately we're concerned about health. And so what has typically been used as an indicator are fecal indicator bacteria. And so a lot of surface water quality standards are based on indicator bacteria like E. coli and Enterococci. These organisms are found in high concentration in animal host feces. However, these organisms aren't perfect. They don't kind of meet all the criteria of what we would say is as a good indicator, but it's really hard to come up with a better alternative, right? So it's really easy to kind of pick at an indicator, but to kind of put forth a better one. And so people are often looking for, you know, improvements to indicators that we're using. But what's also a challenge, one of the drawbacks of E. coli and enterococci are that it's not specific to particular animal hosts, right? So for instance, for recreational water quality guidelines, you know, enterococci is an indicator organism, and enterococci can be found in seagull feces. And so sometimes beach managers are saying, you're making me close down my beach, but we just have a lot of seagulls. You know, this isn't, you know, causing extreme health risk. This is nothing that we can do. And so there has been this growing interest in being able to say, what is the host of the fecal contamination that we're detecting in the environment? And because of that, methods have been developed called microbial source tracking methods that can help distinguish between different host species. And this is helpful because often our management efforts are going to be source specific. So how you would intervene in a situation if the source were coming from humans versus, you know, swine, your management strategy would be quite different. If you're kind of looking at the impact evaluation of different intervention studies, having a target that more closely aligns with the intervention approach that you're using can be helpful. And then also these health risks do vary by source. And so, you know, due to that, you know, beach managers' concern, if you can say, you know, this isn't a human feces impacted beach, even though we're kind of exceeding some of these limits, then that can be helpful for making decisions on closures. I will argue, though, that I think this health risk characterization by source, that, you know, that was a modeling effort. I still think there's room for us to improve how we're characterizing the risk associated with different host species. And so these methods have been developed that take advantage of the diversity of microbes and different hosts, the digestive system of different hosts, and have identified human, ruminant, and avian specific targets for fecal contamination. And we do analyses to assess the sensitivity and specificity of these targets as well. So very quickly in the last few minutes here, I want to just share some of the results that we found when we looked at surface water quality after Hurricane Florence. I'm going to just move quickly through this. And the goal here was to look at microbial contamination. And we're also interested in looking at different land uses and how that might impact contaminant loading. That analysis is still underway because we're also using some you know, advanced methods for estimating flooding extent and using that as a predictor in our geospatial models for microbial contamination. So this was work that was done in eastern North Carolina. We had 44 different sites that we targeted in you know, a wide range of space. And we looked for many different microbial targets, including fecal indicator bacteria E. coli. We looked for a human specific and a swine specific microbial source tracking target. This was a molecular target using qPCR, if you're familiar with that technique. And then we, as part of a group, we also measured these pathogens using culture-based methods, Salmonella, Campylobacter, Archobacter, and Listeria. And we did see a range of E. coli concentrations in the water sample. So we sampled about 1 to 2 weeks after the hurricane made landfall and then 1 month later. And we actually observed similar concentrations of E. coli in the surface water between these two phases. However, we had different sample types as well. So we sampled flowing channels, we sampled floodplain water, and then we also sampled a few isolated ephemeral water bodies. And so the channel samples had higher concentrations of E. coli during the phase 2 sampling as compared to the phase one. And, you know, that could be that there was a dilution effect that happened with the phase one sampling, or it could be that there were unique inputs in phase two because there were other rainfall events that kind of happened between these phase one and phase two, and also the practices. So I think there were some restrictions that were lifted that allowed farmers to kind of drain down their lagoons. And so there could have also been some unique inputs between phase one and phase two. We did see both human and swine targets in the water samples. So 26% of the samples had the human specific target, but it was inconsistent between sites in phase one and phase two, suggesting that there could be unique kind of sources and inputs during this extreme flooding event relative to when there wasn't this as connected water systems. And then the swine target was detected in 9% of samples. And we actually saw that human and swine targets were correlated to each other. So if you detected the human target, you were also more likely to detect the swine target than if you didn't detect the human target. So we saw like a double burden of risk there in our sample sites. We did see a range of pathogen detection, but what was quite surprising was that this emerging contaminant, Archobacter boutzleri, was detected in a large fraction of our samples. Almost 80% of our samples had this target detected between both phase one and phase two. And just to kind of wrap up some of this, we are looking at other targets, including antimicrobial resistant genes and antimicrobial resistant organisms, which is an emerging global health threat. I've also partnered with some people that are looking at emerging chemical contaminants in the water to understand how microbial and chemical contaminants are related in surface waters under different climate scenarios. And then also, as I mentioned, we're interested in understanding relationships with land use and flooding extent, and also contamination as well. Yes, and so just in terms of big picture, I do think that there are ways that we can upgrade our infrastructure to prevent, you know, some of these inputs into the environment. And I think we can innovate with some of our waste management strategies. And there are several farms in North Carolina that are trying out new strategies, and the economics can actually work out. Like if you're using some of the waste as a resource, it can have this double benefit of, you know, resource provision, but also reducing contaminants that would potentially enter the environment. And then also more routine monitoring and thinking about what do we want to be monitoring so that we have better understanding of these dynamics and how it changes over time, and characterizing base flow conditions well if we're really wanting to assess impact. So those are just some thoughts. And you know, this was part of a large research team, and we also had many students that helped with the field sampling efforts. And so yeah, I'm happy to take, I think there's a little bit of time for questions, and you know, happy to continue the conversation offline too. Thank you.
Thanks, Angela. That was awesome. While we wait for folks to post their questions to the chat, I have a question for you. So here you were talking, it seemed mostly about large water bodies. So you talked a little bit about water quality at beaches and bays, along rivers, and after larger rain events. I'm wondering if the same microbial contaminants are of concern for high frequency precipitation events that might produce puddles in some of these low-lying areas along the coast, or street flooding.
Yeah, no, I mean, great question. I think, you know, these are very complex systems. So knowing how your system is going to respond to different types of rainfall events, I think, and what were the conditions prior to that rainfall event as well, I think can impact. But, you know, I was recently doing some sampling near Beaufort on the Newport River, and that system was very, you know, responded very much so to rainfall, you know, even just like 1 to 2 inches. I mean, that's like a a decent, that's a strong, a decent rainfall event. But, we were seeing, it was very clear that we saw a spike of, E. coli and enterococci in response to those types of rainfall events. And so these, it doesn't have to be a large event to mobilize contaminants, but how different contaminants might be mobilized and different systems might be connected depending on the scale of the event.
Okay, thanks for that. Okay, Brad, looks like you just popped one into the chat.
Yeah, so
Yeah.
this is the, yeah, sorry, Brad, you can go.
Well, just, I guess, starting with the first question, is that people actually get exposed to this, the contaminants during, as floodwaters come to get them? Is that the main pathway?
Right. So I don't think we have very well characterized what are the dominant pathways of transmission whenever there is a flooding event. And so it could happen, many different ways. It could be, if people are going into the floodwaters, or, their shoes are going into the floodwaters, and, then their hands are touching their shoes, or, there can be kind of several steps before the exposure event happens. Also so I've been really interested in understanding contamination in soil after floodwaters recede, and how long does that contamination persist? And, because I've worked with respondents and, community members that are concerned about flooding and floodwater quality, and then what's kind of left over after the floodwaters recede. And there's this kind of sentiment that, like, I know I'm not supposed to let my kid play in the floodwater. That's, I get that. But, after the floodwaters recede, there's, trash in my yard or things like that. And so I might go and clean that up. I'm concerned. They're concerned about, critters that also kind of become spread. So there are those concerns, but then they can't see lingering impacts of, the floodwater contamination in their yard. And they don't know, when can I let my kid go back out and play? When can I go back out and garden? And should I garden? And, these types of questions. And I do think that there could be more work to understand what are these impacts? What are the dominant exposure pathways?
Thank you.
Okay, any more questions for Angela? All right, thank you so much, Angela. And thanks for everyone for attending today. Just a reminder that next week we have a talk by Patrick Barnard at the USGS about coastal hazards and federal agency science. And then by Sarah Shoshani at NC State about the long-term resilience and stochastic optimization of these systems, of coastal systems.
In 2 weeks.
Oh, sorry, in 2 weeks at the same time. All right, hope to see everyone there. Thanks for attending. I'm gonna stop recording.